Hi, welcome to Science Hangout. I'm Sadashiva Pai from Science Mission. We have today Dr. James Eberwine, Professor of Pharmacology and Translational Therapeutics, Co-Director, Penn Program in Single Cell Biology at the University of Pennsylvania School of Medicine with us. Dr. Eberwine has published hundreds of research articles and is in the editorial board of several journals and has won several awards and is an expert in single cell analysis. Welcome, Dr. Eberwine.
Thank you for the invitation to join you. I'm looking forward to our discussion.
Can you please tell us about your background, your education, just briefly go over your kind of a history of what you have done so far?
Sure, I'd be happy to. So I was born and raised. I was raised in Ohio, and a small rural town in Ohio, which was phenomenal. You know, my initial science interest came from some high school teachers that I had there who were phenomenal at teaching you how to think about science. I'm afraid so much science education these days is reading and memorizing. But what these two teachers did was they did experiments. They had us do experiments. They had us think about observation and interpretation. And so it was really, really wonderful to be able to have that grounding. And then my real formal introduction to science came when I was a junior in high school. And my parents were kind enough to send me to a National Science Foundation summer program in science. So it used to be that the National Science Foundation would fund high school programs all around the country in various disciplines. So my parents, who believe education is really important, sent me to one on biochemistry in Hartford, Connecticut. And so I spent 2 weeks studying a biochemistry text called Leninger and doing experiments and learning about science. And so it was really a formative experience that really helped me understand that I wanted to be a scientist. And from there, I was fortunate to be able to go to Yale as an undergraduate, where I majored in biochemistry and did lots of philosophy. I did a thesis in philosophy. And from there, then I went to Columbia as a graduate student, then a postdoc at Stanford. And in 1990, I came to University of Pennsylvania as an assistant professor and started much of my independent work at that point, and became the Bobst professor of pharmacology in 1996. So Penn has been a great environment for me. I've really enjoyed it. Among the things that I've really valued in terms of education and thinking about science, and I know Sadashiva, that you're very interested in science education as well, is I was fortunate to be able to start a Cold Spring Harbor course when I was a postdoc on molecular neurobiology. So 30 years ago, 1 years ago, I started the cloning of neurogenes course at Cold Spring Harbor. At that point, there were very few people doing molecular biology in the nervous system. So they asked me as a postdoc to do it. Normally they have professors do it. So I started the course and I directed it for a total of about 15 years off and on. And so in the course, a lecture. And then 5 years ago, I started another course at Cold Spring Harbor on single cell biology. And those courses, I don't know if you've been to one, but they are really phenomenal in terms of bringing among the best students from around the world who have an interest in a particular discipline to the lab to actually do didactic education, learning from papers and lectures, but also hands-on laboratory experience, doing experiments that they might do back in their own lab, applying the techniques that they're learning. So, so I think that's a little bit of my background, some of my interests, and right now, I consider myself a molecular neurobiologist, but also a genomicist, and we do much of our work at the level of single cell analysis in the central nervous system, although we do work in a whole host of other cell types as well. But you'll hear about some of that during my formal presentation.
Okay, then tell us about your lab people, lab grad students, postdocs.
Yeah, no, it's a great question, and one that's critically important to modern science. I've been very fortunate to have great graduate students and postdocs come through my lab. Right now, I have one graduate student and I guess 5 postdocs, 6 postdocs, and 4 technicians. And the thing that I require of these students, whether they're a graduate student or a postdoc or even a technician, is their willingness to think creatively. You know, in my lab, we do a lot of new technology development because the questions that we try to address require that. There's no technique to answer the questions that we're trying to develop. And to do that, you have to be open-minded. You have to be able to understand the technology you're developing. You have to be able to understand when you need to use cobalt or cadmium in your reactions. And so you have to be willing to think creatively and to accept failure, because oftentimes new technologies and new approaches fail. And so I've been fortunate to have almost all the people through my laboratory who are willing to think open-mindedly and to approach science in a creative, fun way. You know, it's such a great time to be able to do science now. There's so many questions that one can finally answer. You can ask any question. You've been able to ask any question you wanted to for forever, but can you actually address those questions? And now we really, really can do many of those. So I think that's really, really phenomenal. And so the people in the lab have been very, very conscientious about, you know, working in that manner.
Your, I think camera is stuck. Looks like basically. Just if you can.
All right.
Yeah.
Oh, I'm not even seeing the camera now.
Yeah.
See if that comes up. For some reason that camera doesn't seem to be coming up now. Let me try it again. Okay,
Here
there we
you
go.
Okay. So one other question is like, how do you motivate them? Like, I know like people like scientists want to have their own motivation, but certainly there is some role to be played by the faculty or the mentors.
You know, I think, again, that's a really interesting question. I think it goes to the heart of your interest in science education as well. And, you know, the motivation, you know, 90% of it has to be self-motivation. You have to want to try to do the work you're doing. You know, my contribution to this is trying to help the people in my laboratory think creatively and broadly about their science and going back and forth. And I seldom say no. It's always, you know, a back and forth discussion of this idea or that idea, and if we want to try to address it, how do we do that? And so it's that interaction which I think helps with the motivation. But I really want people to come into the lab who want to be creative and understand that it's such a privileged position to be able to be in a laboratory, to think about a question for the first time, to be able to address it for the first time. And when you get that data, to be the first person in the world to ever think about it and to be able to try to understand what it might mean. And so that's the type of motivation I want people to have. And it's what I try to help them with in terms of our in-lab discussions.
I know like you are busy with all your work and all those things. So if during your spare time, like how do you spend that? Like what are your hobbies and other things?
Well, you know, that's yeah, you're right. There's not a lot of time. Am I stuck again?
Yeah.
All right, let me see if I can. I switched to another camera. Let's see if this one will work.
Okay.
Yeah, I'm really not sure why it's lagging so much.
Yeah.
let me turn off a few of my other programs.
Okay.
Okay. Yeah, I'm sorry. For some reason, I'm just not getting this.
Yeah, you can go back to the same mode, the camera.
Yeah, that's what I'm trying to do. Yeah, I don't really know what the problem is. Sorry, it just doesn't seem to be coming on.
Oh, really? Wow.
Yeah, I don't know. It's very odd. Let me see if I can try this from my laptop.
Okay.
I should tell you what, why don't I go out of Google and go back in, okay?
Okay.
Okay, let me try that.
Yeah, we had some technical problems, so we should be able to get it going within no time. Here you go.
Okay.
Yeah.
So so yeah, and in terms of hobbies and things that I've done in the past, no, I don't have a lot of time for these things now, but I do like to do woodworking, and so I enjoy doing that. I enjoy going down into my workshop in my basement and listening to music while I make things. I enjoy reading. You know, I read extensively, although not as much as I'd like. Listen to music. Primarily jazz is also one of my loves. In the past, I've done a variety of things. I've played rugby in the past, and I played judo for many years. I was a guide for an organization called International Field Studies for Spelunking, which is cave exploring. So and lots of things like that I've done in the past. But these days, you know, it's a lot of lab work mostly, and not nearly as much time for these other things.
Other things, okay. And what got you interested in single cell analysis, single cell gene expression, and all?
Yeah, no, that's again a great question. So that was when I was a postdoc at Stanford. I was involved with a group that was interested in trying to do analysis of circadian rhythms. Okay. And there's a region of the brain called the suprachiasmatic nucleus, which is involved in regulation of circadian rhythms. We now know that every cell has a circadian clock, but back then we were interested in this one sort of region of the brain. And so we were working in the mouse system, and to be able to generate enough tissue to get enough RNA to do the standard type of molecular biology and library making required the use literally of hundreds of animals, because SCN is such a small area. And so not wanting to do that, not wanting to use so many different animals, I then decided to try to figure out how we could adopt molecular biology procedures to decrease the number of animals and increase the sensitivity and resolution of molecular analysis by working at the level of the single cell. We now spend a lot of time working on subcellular molecular biology as well. But that was really the motivation, with trying to use fewer animals for the research and to try to get as much information from the system as we can.
Yeah. Can you please go ahead and present your work with that as a backlog?
Sure. So what I thought I would do is present some of the work from my laboratory over the last couple of years. Can you see this?
Yeah.
Okay. And I'm going to go through this relatively quickly. I'm sure everyone would rather get off and have a late breakfast or early breakfast, depending upon where you are. And so, but I want to give you sort of a flavor for some of the things that single cell biology has been telling us. So the driving question for my laboratory is really this, and that's how is variation in cell biology important to the functioning of a cell? And so it's illustrated by this Cezanne painting, where the 7 different apples, they all look similar, but they're all also distinct. There's biological variation. And so the question for us is, what does that biological variation mean to the cell itself, to other cells, and to the organism? So normally I would present this in PowerPoint mode. I'm not doing that now because of the limitations of the system, but this is an animated slide. There are lots of animated slides that aren't going to show the animations because of how I'm showing this. But this is just sort of a rationale for doing single cell analysis. On the left, you can see you can see a brain where you can see areas such as sort of the horseshoe shape in the middle, which is called the hippocampus. And on the right-hand side, you can see sort of dark tracks, and that's the cerebellum. So here what we've done is a technique we developed several years ago called in situ transcription, which is cDNA synthesis right in the tissue section. So what we've done is we take a fixed tissue, we put a primer on, that primer will bind to the RNA, and then we can make cDNA right in that fixed tissue. And you can see areas like the hippocampus and the cerebellum where there's a lot of black signal, and that means that there's a lot of RNA there. And so if you go to the upper right panel, you can see 3 areas in that panel that have a lot of white grains. And so this is at the level of single cell. So if we zoom in with a microscope, we can see those 3 cells with a lot of white grains. And those are RNA transcripts for a particular RNA we were interested in. And what this shows us is that those 3 cells all have this one RNA, and there are 100 other cells represented by the brown circles in the picture that do not have this RNA. So there's variation between these cells that have the RNA. There's variation between the cell that has the RNA and the cell that doesn't. And so the question is, how variable are they? And can we develop techniques that allow us to do that analysis? And that's where single cell analysis comes in. And my interest has been primarily the CNS. So there's some facts about the CNS that in the lower right-hand panel that show why single cell analysis is important. One of the things I want to highlight, though, for this audience, which may not be a real neuroscience audience, is that almost all of this work has been done in collaboration with my friend and colleague, Professor Junyoung Kim. Junyoung is a computational biologist, and the distinction I'd like to make is a bioinformaticist is somebody who might teach you French language, whereas a computational biologist is somebody who would teach you French literature. And that's really how Junyoung and I interact with respect to all the work that is done in these particular experiments. We'll do an experiment, he'll help us analyze it. We then go back and do better experiments based upon the analysis. And all of you interested in genomics need to interact with computational biologists in a productive way. I like showing this slide because my son, who is a classicist, helped me do the translation on this. This is a passage from a book that Robert Hooke published called Micrography in 1665. This was commissioned by the Royal Society. It was one of the first scientific bestsellers. 2,000 copies of this book sold within 2 years. And Latin was a language at that time. And when this is translated, what you can see is using the microscope that he invented on the right-hand side, you can see that he was able to look at cork, and he called the pores that he saw cells. And that was the first description of an individual cell, what we call a cell now, as a cell. And so Leeuwenhoek, a couple of years later, followed this with his description of live cells called animalcules. And then over the course of the following 150 years, various people contributed to the development of the modern cell theory. And I would argue that it's an elaboration of the modern cell theory, which gave rise to all of modern molecular biology, including the elaboration of Darwin's hypothesis for natural selection. And without the idea of the cell as an entity in and of itself, I don't think any of these other scientific discoveries would have been possible. An additional rationale for looking at single cell analysis is provided by this slide. So if you look at the left-hand portion of this, you can see 5 blue cells. And if we're interested in the red triangular RNA, if you take all these cells, isolate the RNA from all those cells, which is what we used to do before single cell analysis, all the RNA would be combined in a test tube. And the triangular RNAs are not visible because they're below the level of sensitivity of the technology. On the right-hand side, you can see 5 cells, and each of the cells has differing amounts of the triangular RNA. So if you take all of those cells and homogenize them and look at the RNA, you might think that each cell has 2 copies of RNA. Whereas you can see some cells have none and other cells have large amounts. So single cell analysis allows you to resolve those issues. You can see whether or not a cell has a large number of RNAs or no RNAs. So it allows you to understand that complexity. It's a shame that the animations aren't working for this slide, but you can do single cell biology now for a variety of different cell biological processes. You can look at genomic DNA in a single cell and look at variation based upon single nucleotide polymorphisms. You can look at aspects of chromatin dynamics, single cell epigenomics, single cell transcriptomics, which are what I'm going to primarily tell you about. We've done a lot of work looking at single cell mRNA translation, as we were the first group to show that upon transfection of RNA into cells and into dendrites, that RNA could actually be translated into protein. But I'm not going to show you any of that work today. Single cell peptidomics and proteomics is a developing field, just as single cell enzymology and metabolomics are. And then single cell functional genomics, we spent a lot of time in my laboratory on, but I'm not going to have any time today to tell you about that. But I think it's critically important as you develop all these transcriptomics profiles. There are literally petabytes of data of single cell transcriptomics data and tissue transcriptomics data. And what most people do is they look at one gene at a time based upon that transcriptomics data. So figuring out ways of looking at multiple genes simultaneously is going to allow you to understand what that expression profile is telling you. And so figuring out how to do functional genomics at that level is important. This all gives rise to this idea of cellular phenotype. And phenotype is a much more complex process idea than I think many people think it is. When you think about phenotype, is it function of the cell that you're interested in? If its function is a function of that cell at 10 in the morning or 10 at night, is a function of that cell when it is 2 weeks old or when that cell is 70 years old in a human brain. And so so phenotype is very complex. And I think as we talk about phenotype and elaborate this idea over the coming years, we need to figure out how to describe it. We need an ontology for phenotype that just doesn't exist at the moment. With this said, again, this is an animated slide normally. There's a real move towards high throughput with people doing high throughput sequencing of large numbers of cells, doing high throughput cell biology, and that really is important. You need to be able to generate large amounts of data. But at the same time, it's critically important to do high throughput. It's not sufficient to just do 10,000 cells and sequence them. You have to try to plan that experiment so that you get the most information out of it. And if the most information requires doing 10,000 cells simultaneously at low read depth, meaning you don't detect all the RNAs, then you do the experiment that way. If, however, the experiment requires you to look at transcription factors, which are often low abundance RNAs, then you need to sequence to a much greater depth, and that requires less throughput and higher sequencing ability. So you have to think about the experiment you're doing and really put some thought into it. Again, the next series of slides are animated slides, so you're going to miss that, but I'll quickly flip through these. So when you think about single cell analysis, what you want to be able to do is to detect and quantify the chemicals in a single cell. So this is a neuron, and we'd like to be able to look at the genomic DNA sequence, which is in the pink nucleus area of the cell. We'd like to be able to look at the RNA, which would be in the blue or the green area of the cell, which is in dendrites in the cell soma. We'd like to be able to look at proteins and metabolites. And we'd like to be able to look at all those quantitatively and at the same time in the single cell. So that's called multimodal analysis, so that we can actually not just look at the transcriptome, but look at the transcriptome, the proteome, and the metabolome all in the cell to give us an idea of what the biology of that cell really is. We'd also like to be able to look at organelles in the cell. You have translation occurring in dendrites or in cell soma. Splicing can occur in the nucleus of the cell or the cytoplasm. We were the first group to show cytoplasmic splicing. We'd like to be able to look at transcription and chromatin. That's in the nucleus of the cell. So we need to be able to look at these subcellular structures. Ideally, we'd like to be able to look at all of these simultaneously. So DNA and RNA and protein, we'd like to be able to look at translation as it's occurring, transcription as it's occurring. And basically, if we could do all of this, then we'd have the ultimate characterization of the cell. And while that's not possible today, it will be possible within the next few years to be able to do all of these things. Many of us are working on techniques where we'll marry these different analysis procedures so that we can do this in a quantitative, reliable manner. One of the other things I want to highlight, though, is, as I say this, there is subcellular biology associated with this, particularly with a cell like a neuron. So you have these red rectangular areas where you have dendrites. So that's one component of the cell. And the biology in the dendrite can be different than the biology in the blue rectangle, which is the cell soma. And you can also have different biologies occurring within the nucleus of the cell. So you really do have to be able to not just quantitate these processes and chemicals in the whole cell. You need to be able to look at subcellular sites. And much of my lab is devoted to doing that type of analysis. To be able to do multimodal analysis, you need to be able to figure out how to isolate your samples and quantitate it. And again, I'm not going to go into this in any detail now. But among the types of things that one can do with a single cell, there's low-hanging fruit that's very easy to detect now. And basically everyone who does single cell analysis can do this. And this is actually potential drug targets. We did this work a few years ago now, and it was among the first, it was the first paper describing the transcriptome of a type of neuron called a serotonergic neuron. These neurons are involved in a variety of different functions within the cell or within the organism. And there are maladies of serotonergic neurons, including depressive behaviors, social anxiety, a whole host of other types of neurological psychiatric illnesses that are impacted by serotonergic neuronal function. So understanding these neurons is important to understanding those illnesses, I would argue. So what we did with Cheryl Beck at University of Chop, took a mouse that had serotonergic neurons labeled with a marker that would allow those neurons to be visible. So this is a live slice through a region of the brain where serotonergic neurons are. And so the yellow cells are live cells that are serotonergic. So we can take a patch pipette, which is a glass capillary tube, and go down to an individual cell that has a yellow color and isolate the RNA from that cell. So you can see in panel B, there is the YFP fluorescence. There's a little micropipette or a stick that's poking that cell from the lower right-hand corner around 4 o'clock on the clock dial. And so we can poke that cell and pull the RNA into the pipette. And so we know we have the RNA from a single serotonergic neuron that way. And when we do this, we can sequence it and we can identify a whole host of different RNAs within the cell. And among the RNAs that we're interested in are those GPCRs, G protein coupled receptors, which are receptors on the cell surface of cells. And every major pharmaceutical company in the world has a GPCR program where they try to identify ligands and targets that bind to these receptors. Prior to these studies for these cells, we knew that there were 3 GPCRs on these cells. By virtue of doing the transcriptomics, we now know that each of these cells has RNA from 200 different GPCR genes. So the RNA is present. That doesn't mean the RNA is made into protein, or if made into protein, the protein is functional, but it tells you these cells have the capacity to respond to these particular ligands. And so this data is only generatable by doing single cell analysis. And when we look through these lists of GPCRs, we identified histamine receptor as one that we were interested in, and we were able to show that these cells endogenously were tonically inhibited by histamine. So this was the first proof that there's a tonic inhibition of these neurons by histamine. And so this allows us to identify those receptors and then to do some interesting biology associated with it. And these studies are continuing in our lab and many other labs around the world now. One of the questions is transcriptome variability important to cellular function? And so I started the presentation that way. We have lots of ways of looking at this, and again, the animations are not going to come through on this, unfortunately. But if you look at this slide, along the top you see a purple line called for serotonergic neurons, a light blue line for cortical pyramidal cells, a red line for cardiomyocytes, a brown line for brown adipocytes, and below each of those lines are columns. In each of those columns is a single cell from that cell type. And what you can see is the neurons, serotonergic neurons, and the hippocampal pyramidal cortical, the cortical and hippocampal pyramidal cells express neuronal markers, and those are the yellow and brown areas below those lines. But they also express some cardiomyocyte markers that you see as the red line below that. The cardiomyocytes, which are over a little bit over further right, if you look down those columns, you can see the cardiomyocyte markers come up high, but also some of the neuronal markers are coming up. And if you look at all the cardiomyocyte markers, or you look at the neuronal markers, you can see that from column to column, they're not the same. They're different gradients of color. That means there are different amounts of RNA for those, and that tells us that the cells are variable in the amount of RNA expressed. So even for a particular cell type, like a cardiomyocyte, there's variability in the markers that are known to be present in cardiomyocytes. And so one of the things that this highlights, and everyone doing single cell transcriptomics, no matter what cell type, is seeing this variability now. But one of the things I want to highlight is we used to describe neurons versus another cell type called astrocytes, as astrocytes would express something called GFAP, which is a marker of astrocytes, and neurons would express neurofilament, which is present in neurons. It turns out that I can find GFAP RNA in some neurons. I can find neurofilament RNA in some astrocytes. And these markers, all the work that's been done in the past has been right, but it's been the level of sensitivity. If you do immunocytochemistry or you grind up a tissue, you don't have single cell resolution, and you're going to miss those lower abundance RNAs that are present. So there are very few individual cell type specific genes. One of the things that is important to take note of with respect to this is as you develop transgenic models and try to develop a cell type specific promoter, many of those promoters, when you go to the level single cell, are expressed in all alternative cell types. So you have to be extremely careful, I think, as you develop those models these days. There's a lot of other biology that one can develop, and this is all published, so I'm not going to go through this in detail as again, I don't want to spend a lot of time. One of the things I do want to mention, though, is when you look at gene RNAs, gene-transcribed RNAs in cells, there's a certain gene model that people always look at, where the 5' is, the exons, the 3'M. But what this data shows, and this is all published, so you can look the paper up, is that even within a particular cell type, like such as cardiomyocytes or pyramidal neurons, you can find different 3' ends for the RNAs. So the same gene, but different 3' ends. And the 3' end of the RNA can have impact in terms of how stable an RNA is, or even the function of the RNA, because the 3' end can have different exons. And even within an individual cell, you can have different 3' ends utilized. So this is telling us something about the molecular biology of the cell that we did not know before single-cell biology. And I think is, again, going to be important for how we develop the genomics of the nervous system and cell types. Of course, we'd like to do multiple biology simultaneously, and we want to be able to do this in human cells. So while we do a lot of mouse work, and it's a great model system, we've also worked in the Drosophila system with Anita Segal and other systems with other people, including some very interesting work in quail with Rusty Lansford at USC. We'd really like to be able to work in the human system. Even though we work with model systems, there's no cell like the human cell, and many things that you see in a mouse cell, which is a standard model system, are not going to be the same in the human system. So we need to be able to look in the human as well. So we were fortunate to be able to work with a neurosurgery group here at Penn, and we have an extensive collaboration so that at the time of neurosurgery, tissue that is removed, such as for a glioblastoma or interventional neurosurgery for epilepsy, as those manipulations are done and the tissue is removed, we take that tissue and it comes to our laboratory. And what we've been able to do is we were able to do the first human neuronal adult culture from these tissues. So we can take live human brain tissue, dissociate the cells under certain conditions, and culture adult human neurons from tissue that was isolated from 70-year-old patients. And so, again, this paper was published this past year. And these are some human neurons that we have in culture. And if you're going to develop drugs and try to understand human neuronal biology, you have to work with the human system, because there are just some things that are going to be unique to these human cells. And so, so we've been able to do this. We can record currents from these cells and do a lot of biology on these cells. Here, maybe this animation will show. Here we have human neurons in culture, and we are stimulating with various types of drugs, such as glutamate or with potassium and other types of markers that one of these will show, that cause calcium to come into the cell. And so the lower right-hand panel shows us cells that are ATP responsive or potassium responsive or glutamate responsive. And so now we can go to those individual live human neurons and pull the RNA from those individually responsive cells into the pipette and try to understand how the physiology and responsiveness to these drugs correlates with the transcriptome of the cell, to gain biological insight. In addition, we've been able to learn how to culture these neurons long-term, and in fact, we can actually get neurospheres developed from adult human brain. So what you can see here is after 3 months in culture, we have neurospheres that are developed. And from these neurospheres, new neurons and astrocytes actually are generated, even again, from the 70-year-old adult brain. And this is actually from an area called the hippocampus, I'm sorry, from the cortex, where people had not thought that this was a tissue where this could be done. And so this gives us a lot of hope for the ability to manipulate the human adult central nervous system in a way that will allow neurons to regenerate and perhaps reconnect and recapitulate the function that some disease cells might actually have subserved. In our cultures, we have neurons, we have astrocytes, we have a variety of different human cell types. Again, you can look this paper up. I'm not going to go through this in detail. This is just the transcriptomics signatures of these cells. And when we have these transcriptomes, we can then try to understand how variable these cells are from one another. And again, this is all published. This is just a statistical way of looking at these 5 different cell types and showing how different they are from one another and how similar they are to one another within a cell type. And we can also look at RNAs that are not mRNAs. This is actually, these are long non-coding RNAs. These are regulatory RNAs that serve a variety of roles within cells. And some of these RNAs seem to be cell type specific. If you look in panel C, the first link, which is 1314, you can see way out at the right-hand side of that, a red arrow pointing to the line, and that's link RNA that's greatly enriched in astrocytes and present at very low abundance in these other cell types. And there are other RNAs, such as the lower one, that seems to be present at relatively high abundance in all cell types. So again, the transcriptomics gives you insight into specific ones that one might want to look at. You can also look at pre-microRNAs. A microRNA would not be isolated in our procedure, but a pre-microRNA would be, and we've been able to identify some pre-microRNAs that seem to be relatively enriched, say, in endothelial cells, such as MUR145, and other ones that are distributed throughout cells. And so this is a particular cell type that a lot of us, or RNA type that a lot of us are interested in because, again, of its regulatory role in modulating RNA expression and protein expression. So what I've shown you mostly is RNA from cells that are dispersed in cell culture. What we'd really like to be able to do is look at RNAs in their natural microenvironment, where a neuron is connected to other neurons, and astrocytes and endothelial cells are interacting with those neurons, where we don't take the cell and disperse it from the brain. We want those connections to remain intact. So some of the early work I showed you, where we took the serotonergic cell RNA, that was from a live mouse brain section, and we found those cells through fluorescence microscopy. We took a pipette and went through the tissue to isolate the RNA. But we wanted to do and develop a procedure that was not invasive like that. We wanted to develop a procedure that didn't require the pipette to isolate the RNA. So working with my chemistry colleague, Ivan Mohovsky, who is a professor in chemistry, we developed a procedure called TIVA. And what TIVA does, again, this is an animated slide, so I'm afraid that's going to be lost on you, is it's a way of isolating RNA from live cells. And what this slide is intended to do is to show you how you isolate RNA from pools of cells in tissue, single cells in culture, single cells in fixed tissue, or single cells in live tissue. And so I'm going to just show you a little bit of some of the TIVA work. So the TIVA work is based upon this particular compound. So this is a complex compound that we synthesize in chemistry. And the idea behind this compound is, if you look at it, on the structure you see a CPP listed sort of in the middle of the slide on the chemical structure. CPP is called a cell-penetrating peptide. And this is a peptide that, when connected to this molecule, will carry this molecule into any cell. And once it's inside the cell, it sits there. And it sits there because of the chemical structure. You see a poly U, which is in blue, and two short stretches of poly A. Those poly A's bind to the poly U's through hydrogen bonding. Those are nucleotides. The poly A's are separated with that red chemical. That's a photoactivatable compound. And the U's and the A's are separated from each other by that same photoactivatable chemical compound. Beside the blue U's on the left-hand side of it is a fluorescent molecule called Psi3. And beside the green A's in the bottom, there's a fluorescent molecule called Psi5. So when this molecule is in the cell, the Psi3 and the Psi5 are very close to one another. We have what's called a Fret signal. So you can see them interacting. And then when we hit this compound with light in the cell, the red molecules between the blue and the green U's and A's, and then between the green A's, break apart. And when those break apart, then the A's fall away from the U's. The U's can then bind to the A-containing RNAs in the cell. The Fret signal is lost. So we now know we've activated this compound, and the TIVA compound basically will bind to the endogenous RNA when this is hit with light. And this is in live cells in real time. And then what we can do is we can grind up that tissue, and you can see next to the top fluorescent molecule, there's a molecule called biotin. So the biotin TIVA U stretch will bind to the poly A plus RNA in the cell, and then we can isolate the poly A plus RNA TIVA complex by using something that will bind to the biotin. So again, this animation is not going to show up, unfortunately, but this is just an animation sort of showing this going to the cell, activating it in the cell. It basically sits in the cell until you activate it. You then lose your Fret signal, and you can then isolate RNA from that live cell. And this paper was published about 2 years ago now. This works excuse me, in a number of different cells. Red is a loaded cell with the TIVA compound where you see the Fret signal. When we hit it with light to activate it, the Fret signal comes apart, and we now have a green signal. So now that activated TIVA compound combined to RNA in that green cell. And we were able to show that we could do this for RNA in the mouse slice preparation, hippocampal cells, and there are a number of different features of this that I think might be interesting. Again, this is an animated slide, so unfortunately it's not going to show up. But one of the things I ask my audiences is, say you have a hippocampal cell in dispersed cell culture, or you have a hippocampal cell in the live hippocampal slice. Which of those cells is going to contain more RNAs? And it turns out that the cell in the hippocampal slice has about 9,000 RNAs expressed. The cell in dispersed cell culture has 12,000 genes expressed. So when you remove the cell from the natural microenvironment, you get more genes expressed. And what that's telling us is the natural microenvironment serves as an inhibitor of gene expression. It suppresses the genes that are expressed from that hippocampal cell. And so that's one way the central nervous system controls the function of these cells. And again, you know, there's a variety of things that we could discuss, but since I can't do the animations, I'm going to just skip over it. We have, of course, applied this to human tissue. So this is actually human neuronal tissue from a patient presented with hydrocephaly. This is a piece of the tissue that was surgically removed. And we can take this and do live slices through the human brain tissue, activate an individual cell, and do the transcriptomics. And this has been a great collaboration with the neurosurgeons at Penn, and it's been phenomenal to be able to work with Sean Grady and his team of neurosurgeons. Not only can we look at the RNA in individual cells, but we can look at regions of the cell. So one area that's been really important and interesting to us is being able to look at RNA in dendrites. And so I highlighted the dendrites in one of the early slides. But the dendrites receive input from presynaptic neurons, from the axons. And when an axon abuts onto a dendrite, you form a synapse. And so that synapse, where aspects of something called learning and memory occur. Every neurological disease known to man has atrophying of dendrites and axons. And so this is a critical subcellular site that's important for how neurons interact with their neighboring cells and environment. So we wanted to look at the RNAs that are present within those subcellular regions. We knew the protein synthesis was important in these areas from the original work of Ozzie Stewart. Then we followed this up by showing that RNAs can be translated. But to look at the transcriptome in these individual cells, what we did initially was, and we have several papers published on this, is in our cultured neurons, we would remove the cell body, and that leaves the fine hair-like structures, which you can see in the right-hand panel. And then we use these glass capillary tubes, basically to suck up an individual isolated dendrite. And then we can do transcriptomics on that dendrite and identify the RNAs that are present there. And again, we have several papers published on this. But using the TIVA compound, excuse me, what we'd like to be able to do is look at dendrites in the live slice preparation, where these neurons, the dendrites, are connected to other neurons. So here you can see in the mouse slice preparation, we activate the TIVA compound in multiple dendrites in the second set of panels, and an individual dendrite in third set of panels, and at an individual dendritic spine, which is where an axon abuts to form a synapse, in the lower panels. And this is possible because of the resolution of light. Now, light will allow us to focus its beam at an individual area of a cell, activate the TIVA compound, and the only RNA that will be isolated is in that green area in that activated cell. So we look at RNA in these different subcellular sites now. And this is just an example of uncaging in different areas of the cell to show that this can be done in the human case. And there are various bits of data that I'm just going to skip over with respect to this. But in some more current work, what we've been doing is looking at human cerebellar Purkinje cells. These tissues come from glioblastomas that are pervasive in the cerebellum, unfortunately. And what you can see is in these live cerebellar tissue slices that we can activate the compound. So if you look, say, at VRU 295, you can see Cy3 and Cy5 cage in that one set of neurons. And in the lower panel, you can see that when we uncage it, the Cy3 fluorescent signal increases, showing us that the fret's been lost, and the Cy5 decreases. So we've activated in that one dendrite in the live human slice preparation in a cerebellar Purkinje cell. And we can now isolate the RNAs. We've done this, and we've looked at libraries of We sequenced many of these, and we can identify the RNAs that are present in these different subcellular sites. So in this top set of panels, in that one dendrite from that human cerebellar Purkinje cell, we have 533 RNAs we isolated from that individual dendrite. And you can see the numbers that we identified in the others. And interestingly, some of these are the same as what we identify in the mouse or the dispersed human cells, but many of them are different. And what that's telling us is the connections and the interactome is really important for elaboration of the functional dendrite in these systems. So we're really excited by these data. The last bit of data that I want to show you is some more recent work, or some ongoing recent work. Again, this is an animated slide. But when we're interested in cell biology, what the cell is essentially the emergent property of the function of the cellular structures or organelles. So a cell is more than the sum of the parts, is what this means. But the parts are necessary to create that functioning cell. So what we'd like to be able to do is not only look at the RNA or the biology of the whole cell, we'd like to be able to look at the RNA in the Golgi apparatus, so the RNA in the cell membrane, or the RNA in the lysosome or the mitochondria. Or we'd like to be able to look at other aspects of the genomics in these individual subcellular sites. And by doing that, then we hope to understand the subcellular constituents and how they work together then to create the emergent property of a cell's function. So we just published a paper about, I think, a month and a half ago now, where we looked at individual single mitochondria, where we isolated individual mitochondria from live cells, live neurons and astrocytes, and then we sequenced the genomic DNA from that individual mitochondria. And the idea is we want to look at mutations in the mitochondrial DNA between cells, but then also within a cell, because an individual cell can then literally have many hundreds of mitochondria. Are all those mitochondria the same, or are they different? If they're different, how different are they, and do they contribute to the functioning of the summation of the function of the mitochondria and the functioning of the neuron? All of us think of the mitochondria as a powerhouse of the cell, but they do many other things as well. So this just shows mitochondria in astrocytes and neurons. Just one way of, this is a live image, and one could, and again, the animations aren't going to work, unfortunately, but you can see each of the red spots in the sort of the dispersed flat cells is an individual mitochondria, and then sort of the green neurons, you can see mitochondria all around the cell soma, and some of them in the dendrites. And so these mitochondria move within the cell, they respond to different types of stimuli, and are actually quite dynamic. This is actually, let me see if this will play. Here you can see live mitochondria moving within a cell, and some of them move quite a distance, some of them fuse with other mitochondria, and so it's really a phenomenal biological process that's going on. And we can isolate an individual one of these mitochondria, and many of these from the same cell, to try to look at how they might function. The mitochondrial genome is about 16,000 bases long, and so that's what we're sequencing. We're sequencing the genome of these mitochondria. It's about 16,000 bases. And there are mutations associated with these different bases that are important for aspects of mitochondrial disease. And many of these mitochondrial disease have a low prevalence. They don't affect a lot of people. Many of them are categorized as orphan diseases. But I think it's arguable, and most people would argue this now, that mitochondria are involved in many other illnesses. So whether a direct mutation is associated with an illness, dysfunction in mitochondria almost certainly is associated with every neurological illness. You have decreased mitochondrial function, making a cell more likely to be responsive to some type of bad challenge or some type of dysfunction. And so how that might be impacted by mutations in the mitochondrial genomic DNA is a critically important question. One of the complications of this is each mitochondria can have one genome or 10 or more genomes. So when you isolate the mitochondria, you might have 10 mitochondrial genomes. So looking at individual mitochondrial genome variation becomes quite complex. So this is a difficult set of studies. Again, you can look the paper up. I'm not going to belabor you with the issues associated with it, but suffice it to say that we figured out how to do this type of analysis so we can look at these mutations and look at single nucleotide variation. The way we do this is you can label the mitochondria with live dyes. So this, again, these are live mitochondria. You can see the fluorescence in these mitochondria. And you can, if you look at A, B, and C, you can see the red arrow in panel B pointing to the mitochondria. And we can take a pipette down to that individualized cell and pull that mitochondria into a pipette, independent of the other mitochondria. So it's brute force isolation of this mitochondria. And we can then sequence the DNA from that individual mitochondria. The way we've done this initially is through this particular PCR protocol. Again, you can look it up. But we have other ways of doing this now that I think are even more robust and more exciting. But when we do this, we can look at the distribution of mitochondria mutations across the length of the mitochondria and between cells. And again, this is all described in the paper. And so we do find variation. So all mitochondria within an individual cell are not the same. And the extent of that variation is actually quite intriguing and exciting for us to be able to investigate. This is actually, I love this slide. This is actually a human adult neuron that had been in culture for about, I think, 4 months. We stained this with a dye from mitochondria, and we could go into the dendritic area of this cell and pull individual mitochondria from each of these boxed areas. And so we can take these individual mitochondria from this mitochondria neuron and sequence it. And so we've done 10 from this individual cell. And again, we can look at mitochondria single nucleotide variation across the length of the mitochondria between cells and within cells. And again, if you're interested in this, you can look the data up. But there are different types of mutations we've identified. We've identified the frequency of these mutations. And these mutations exist in a number of different genes involved in different biologies, including ATP generation, energy utilization, import and export of chemicals within the mitochondria. And we think these mutations are going to be important for how these mitochondria then interact with the nucleus. So there's complex communication between the nucleus of the cell and the mitochondria of the cell. And these mutations are allowing us to try to investigate that in a systematic way. This leads us to following up on the idea of others. So this is not our idea, but there's been this idea in the mitochondrial field that mutations build up over time and reach sort of a critical disease threshold. And when it reaches that threshold, then disease can then manifest itself. And so by doing single cell mitochondrial sequencing and looking at all the mitochondria from these individual cells, I think we're going to be able to understand these mutational loads in a way the population studies would not allow us, and trying to understand the impact on aging and other types of mitochondrial disease-associated biologies. One of the intriguing things that came out of these data is it looks like the mutation frequency is different between mice and rats. I'm sorry, mice and humans. So that I think the ways in which these mutations are generated and evolved highlight the need to actually be able to do these experiments in humans. My next, I have 3 more slides, and I like showing this. Again, this is an animated slide, but I just want to highlight, I've gone through a lot of things very quickly, but it's the cell dynamical states that result from multimodal biology, whether you look at the RNA, the protein, the metabolome, whether you look at transcription or translation, or, excuse me, RNA degradation. All of these go together to create the dynamic cell. And I've made it look easy, but there's a lot of failures associated with biology that I described as well. And so one of my favorite short story authors is Alice Munro. And when she won the Nobel Prize in 2013, she gave an interview to the New Yorker, and this statement was in this interview. The complexity of the things within things, just seems to be endless. I mean, nothing is easy, nothing is simple. And that was true for Alice Munro in terms of the complexity of her writing, and it's true for any of us looking at the complexity of single-cell molecular biology and genomics. It's not simple, but you need to generate the data so that you can then ask informed and interesting biologies and questions of those biologies. And this is only going to, not only going to be possible in dispersed cells, but also in live brains, where you can look at different areas, such as these dendritic areas, where you can see dendritic spines in this region. And so you need to be able to develop techniques that allow you to do this in a multimodal, multicellular way. And then finally, these are some of the people that have been involved in this work. And so Jackie Morris did the mitochondrial work with Waju, and Jennifer Singh did the serotonergic work. Kevin Miyashiro was involved in aspects Actually, I didn't discuss any of Kevin's work. Jae-hee Lee has been involved in all aspects of the TIVA work, and the work of the other people I didn't describe today. Jae-Yoon Seo is an associate professor in pharmacology. He's a biophotonics expert and involved in all the imaging components of what I described. Junyoung Kim is a computational biologist and colleague, and people in his lab and my lab work constantly together to try to improve our biology and to understand the biology. It's been a great collaboration. And we've been fortunate to be able to work with a whole host of collaborators, both within Penn and at various universities around the world, to try to utilize their insights and biologies to try to understand the types of questions that we can address. So that's really it for the presentation. That's what I wanted to try to describe in terms of this type of work. There's a lot of other stuff that could be done. Oh, I don't know what's happening.
I think you shared, I guess. Just unshare it. Yeah, here we go.
There's a lot of other types of biology that can be done. There are a lot of exciting things that I didn't describe that are ongoing now in my lab and other labs. And I just encourage all of you to follow the field. If you're not doing single cell biology, I'd encourage you to do it. It'll help advance the science that you're doing. There are all types of initiatives, funding initiatives around the country and around the world. One of the things I do is I'm on something called the Multi-Council Working Group, which is an advisory committee to the BRAIN Initiative. There are, I think, 12 of us on this committee, and there are constantly RFAs going out for single cell analysis, both from the genomics to the functional level. And so I'd encourage you to look at that. There's a human cell mapping hub map from NIH. There are all types of private initiatives. There's the International Cell Census Initiative, and there's a lot of opportunity to take your science to the level of single cell if that level will enhance the types of biology and help you address the questions you're interested in addressing. And of course, never hesitate to email me or call me should you have any questions about the presentation or any aspect of this work. So thank you, Sadashiva.
Thanks, Jim. I really appreciate it. It's a really wonderful overview of whatever the things happening in your lab and around the world on single cell analysis. So I have a couple of questions, maybe. One is regarding like you have looked at various RNAs in different areas. The question is like there is a more or less like that is what like the people are looking at right now. Scientists are looking at this RNA is more, or like you'll be able to with this single cell genomics you'll be able to see very low amounts of. But does it mean sometimes like this low means like see it all depends on like how they are transcribed. So sometimes like even if there is a bulk amount of RNA, they are transcribed very slowly. So end product is basically the proteins which control the various cellular aspects. So like does it help to do the proteins along with this to get a better understanding on what's happening in the cell?
Yeah, so a couple of answers to that. Again, that's really a great question, and one that we all of us in the field of single cell analysis are interested in. So transcription is thought to occur in bursts, so that you have the genes bursting in RNA for a short time period. And that's thought to occur for most genes. I'm not sure it occurs for all genes, but for most genes. And where you get most of the regulation in terms of production of protein is that translational regulation. Mm-hmm. So what you're suggesting is that the protein levels may not necessarily parallel the RNA levels. And that's exactly right. So all of us look at RNA levels. All of us infer RNA abundances from the RNA levels. I think there's some caveats to that. I think one has to be careful how one quantitates their RNA-seq and keep those numbers sort of as sort of squishy numbers, if you will, because there's a lot of amplification associated with RNA-seq. There is some single molecule analysis now being done that doesn't require amplification, but that's not routine at the moment. But yeah, I think when you have an RNA present in the cell or in a tissue sample, that does not mean the protein's necessarily made there. A lot of people make the argument the protein's made. And in fact, it may or may not be. It just tells you the cell has the capacity to make that protein. So if you can couple it with protein analysis, that will always provide you more insight into the biology of the cells. Excuse me.
And the other question is the dendrites, like dendritic analysis you had done. Like dendrites are sometimes so big, like the different RNAs expressed at different levels and different areas within a dendrite. So have you been able to interpret that or look into that?
Yeah, it's exactly right. And we've done a lot of it. We haven't published any of this yet, but we've looked at different branches of individual dendrites, different areas of the dendrite, and different subcellular compartments within the dendrite. I won't describe it in detail, but yeah, that's exactly right. That follows on the beautiful work, I think, of Ken Kosick and others who have looked at this type of thing in the past. And so, yeah, we've been able to isolate those different regions and analyze them, and are working to try and understand what it's telling us about the biology of neuronal interactions now.
And lastly, coming to a latter portion of your talk on the mitochondria. So within a cell, there are hundreds of mitochondria, like you find all these gene mutations. So is there any number of, like, is there any percentage of mitochondria which are like defective or like mutated, or which are not, like anything has been done?
Yeah, so it's really an exciting question to ask. And quite frankly, we don't know the answer yet. So it's an area that we're keenly interested in. There's, we actually were able to show this, but we were not the first to publish on it, unfortunately. But it turns out that mitochondria can move from an astrocyte into a neuron.
Yeah.
So you're transferring genomic information from one cell to another cell. And so I think there's a lot of there's more specificity to mitochondria than I think we ever thought before. The extent of that specificity is unclear at the moment to us, but we're actively looking at that and trying to understand what it might mean. So yeah, I think it's a great area for future investigation.
So with that, I think we will wind up this Hangout. Thank you so much, Jim. It's really nice to have you on, and we'll look forward to more of your work and more of these Hangouts. Feel free to get in touch with me if you think there is some interesting result. We can just do a Hangout anytime.
Thank you. And I want to also thank you for doing this. I think science education and promoting science across different disciplines and platforms such as the internet like this is just critically important. So thank you for your effort as well. Take care.
Thanks.
Bye.
Bye.